Welcome to today's Explainer. I am absolutely thrilled you're here, because today, well, we're exploring a journey that sounds like it was pulled straight out of a science fiction novel. But the craziest part, it is entirely real. We are looking at the quiet revolution of biological hydrogen. Over the next few minutes, we're going to unpack an incredible story that spans decades, literally going from a forgotten university basement experiment all the way up to modern AI-driven clean energy solutions.
So I want to hook you immediately with a core mystery. What if we could produce clean fuel from organic matter using only microbes? Just take a second to really think about that. This isn't a new thought. It's actually a radical idea decades in the making. It's the concept of taking what we consider garbage, absolute waste, and turning it into pure, clean energy simply by trusting microscopic life to do all the heavy lifting.
Section 1. A Vision Born in Crisis.
Okay, to understand where biological hydrogen is heading, we actually have to time travel back a bit. We're going to the 1970s. Everything kicked off with the 1973 oil embargo. That event completely shook the foundations of industrial energy worldwide. And while most scientists immediately started looking at solar and wind, a tiny group at the Technical University of Berlin asked a totally different question. In 1977, Professor Matthias Reuss founded the chair of bioprocess engineering at TU Berlin. He and his colleagues, including Professor Peter Neubauer, began experimenting with anaerobic bacteria. Their goal was almost unthinkable at the time. They wanted to extract hydrogen directly from biological solutions. It's wild to think about how far we've actually come since then. Back in the 1970s, researchers were working with super rudimentary tools. We're talking liquid substrates, basic microbial cultures of Clostridium and Enterobacter, and just simple sealed glass reactors. They didn't have genetic sequencing. They had absolutely no real-time sensors. They literally just watched hydrogen bubbles rise in a glass tube, which they kind of poetically viewed as whispers of a new energy future. Now, fast forward to the 2026 reality, we're talking AI models, continuous membrane reactors, and omics-guided engineering. The early pioneers definitely didn't have our modern tech, but man, they had curiosity and conviction.
Section 2. The Long Silence and Revival.
So the crucial piece of the puzzle here is understanding why the field stalled out. The reasons were largely economic and, political. From 1977 to 1980, a researcher named Waldemar Fort worked on technology transfer. He was desperately trying to get industry interested in these TU Berlin findings, but it was a massive uphill battle. The cultures were unstable, the yields were pretty low, and then, fossil fuels simply became cheap and abundant again. Funding dried up practically overnight, and global attention completely shifted over to things like ethanol and biodiesel. The reactors were literally packed away.
But here's the thing. The science never actually disappeared. The 2000s sparked a massive revival. The really pressing modern realities of climate change, plus a renewed panic over global energy security, brought biological hydrogen right back onto the world stage. The very ideas that got locked away in the 1980s were suddenly exactly what the 21st century needed. The field was breathed back to life, armed with entirely new advances in microbiology.
Section 3. How biological hydrogen works.
Let's get into the nuts and bolts. It all starts with something called dark fermentation. This is the process of using anaerobic bacteria, specifically thermophilic, which means heat-loving, and mesophilic, or moderate-temperature bacteria, to break down organic waste and release hydrogen gas. And crucially, it happens entirely without the need for light. It completely thrives in oxygen-free environments. Today, decades after those initial TU Berlin experiments, dark fermentation is still the absolute backbone of biological hydrogen production.
3.5 moles of hydrogen. I really want to emphasize this specific number for a second, because it represents the theoretical limit of hydrogen you can possibly get per mole of glucose. Thanks to incredible modern advancements like omics-guided engineering, which basically uses immense amounts of biological and genetic data to optimize the microbes, the very best laboratory systems today are actually approaching this ceiling. Now, sure, real-world yields outside the lab are typically lower, but this number, it proves the immense untapped potential of what these microbes can actually do.
To get closer to that maximum efficiency in the real world, modern engineering uses a cascade approach. Step 1 is that dark fermentation we just talked about, breaking down the initial waste. But then we move to step 2, photofermentation. Here, things like purple non-sulfur bacteria, such as Rhodobacter capsulatus, and cyanobacteria are introduced into the mix. They actually use light to consume the organic acids left over from step 1, squeezing out even more hydrogen. Finally, step 3 is the hybrid output. This combines these processes, or uses anaerobic digestion, to maximize the total hydrogen yield and even produce secondary biogas. It's honestly an incredibly elegant biological assembly line.
Section 4. Modern bioreactors and AI.
Today, we aren't dealing with simple glass jars anymore. We have highly controlled environments. Continuous stirred tank reactors, or CSTRs, along with UASB upward flow systems, completely dominate large-scale fermentation. Then you have packed bed reactors, which are specifically designed to improve microbial retention and keep those delicate cultures stable. And finally, membrane bioreactors. These allow for the continuous extraction of hydrogen, which is absolutely brilliant because it reduces product inhibition. Basically, it means the bacteria don't slow down their production just because the gas is building up around them.
But you know what the real secret ingredient is? The thing making all these modern bioreactors so insanely efficient? It's artificial intelligence. Real-time sensors are constantly feeding data into machine learning models, and these AI models can instantly predict the absolute optimal feedstock blends, perfectly optimize the pH and temperature, and even calculate precise hydraulic retention times, which means exactly how long the liquid needs to stay inside the reactor. This takes all the guesswork right out of the microbiology, vastly reducing the energy input required to run the whole operation.
And the result of all that incredible AI optimization is this highly anticipated number right here, $1.20 to $1.50. This is the reported cost estimate per kilogram of biohydrogen in 2026. This is exactly why AI-driven economics are a total game changer for the industry. By making the process predictable and super efficient, the cost of generating clean fuel from biology is dropping into an incredibly competitive zone.
Section 5. The circular hydrogen economy.
Think about the everyday stuff we just casually throw away. Beet pulp, rice straw, corn stover, whey, even spent coffee grounds. These are the actual organic wastes being used as feedstocks right now. Biohydrogen takes agricultural and industrial waste and turns it directly into fuel, and the leftover digestate can even be used as fertilizer. It requires almost no new land and creates minimal food competition. It is a perfectly closed loop, taking the things we throw away and magically turning them into power.
And this paints an incredibly tangible picture of our future. Right now, in rural communities, small-scale bioreactors are actively being tested for on-farm hydrogen production. Just imagine a farm using its own agricultural waste to power its tractors, heat its greenhouses, and refuel fuel cell vehicles right there on the property. No massive pipelines, no global shipping. We are talking about decentralized power generated locally that could totally reshape our energy independence.
Section 6. Challenges and a lasting legacy.
Of course, we do have to impartially review the engineering and economic challenges, because the system isn't perfectly seamless just yet. Biohydrogen still heavily competes with cheaper, established hydrogen that's produced from natural gas. We still absolutely need higher real-world yields. Those microbial communities, they must be kept stable over very long runs, which isn't easy. And frankly, the gas has to be collected, purified, and stored at a cost that actually makes sense for a small community. But notice something here. These are now challenges of engineering and economics, not basic feasibility. And that is a huge distinction.
I absolutely love this quote from our source material. That conceptual shift, more than any single result, is what later generations inherited. What made the early work at TU Berlin endure wasn't their immediate hydrogen yields. I mean, let's be honest, those early yields were pretty modest. The true legacy of the 1970s was the conceptual shift of trusting microbial metabolism to do chemistry that human industry just couldn't replicate. They successfully reframed organic waste from a nuisance into a highly valuable feedstock.
Which brings us to our final thought for today. How might decentralized, waste-to-fuel energy reshape your own community? Imagine a future where your town's literal waste powers your town's transport. A technology that was once confined to sealed glass jars in a university's basement is now running pilot plants across continents. I want to thank you so much for joining me on this explainer today. Keep asking questions, keep looking for the structure in all this complexity, and I'll see you next time as we continue to decode the fascinating world around us.